Welcome to this course titled Essential Biostatistics for AgChem and Bioscience R&D, a practical guide for decision makers and experimentalists. In the fast-paced world of research and development, data-driven decision-making has become the cornerstone of success. The ability to analyze, interpret, and communicate data effectively is crucial for professionals involved in the field of biostatistics. However, for many non-mathematical individuals, the intricacies of statistical concepts and techniques can often feel like an impenetrable maze. Whether you're a researcher, clinician, pharmaceutical professional, or any other non-mathematical R&D professional, this course will equip you with the tools to navigate the world of biostatistics with confidence. Our approach focuses on providing practical insights, rules of thumb, and real-life examples to facilitate your understanding and application of key statistical practices commonly used in the field. This course aims to bridge the gap between statistical theory and practical application. Our primary objective is to empower participants with a comprehensive overview of essential aspects of biostatistics for trial planning and data analysis without overwhelming them with complex mathematical formulas. My name is Harry Teicher. I'm a crop protection agrobiologist who holds a PhD in plant biochemistry and molecular biology from the University of Copenhagen, and I'm a strategic research and development consultant to the crop protection industry. My experience includes 20+ years in corporate and startup formulation agrobiology, with an emphasis on the agrobiological aspects of formulation bioavailability. During the more than 20 years I spent in crop protection research and research management, I've come to realize that while I and many of my colleagues had followed the same courses in biostatistics, very few of us could apply the methods we had learned or analyze and understand the data generated without repeatedly referring to our textbooks. In addition, it soon became apparent that a subset of the methods we had learned were sufficient to analyze the majority of our trials. To address these issues, I prepared a series of internal courses in these most used quick and dirty techniques, approximations, and rules of thumb for my laboratory colleagues, and these courses proved so popular I was asked to compile them as a printed book. The result is presented in this course, aimed at scientists and research workers not necessarily trained in mathematical theory, but who have previously followed a course in biological statistics and who need a readily accessible overview on how to plan, implement, and analyze day-to-day experiments without access to a dedicated staff of statisticians. It contains few calculations, the how of biostatistics, but instead provides an overview of the why. What is it the numbers are telling us, and how can we use this to plan trials, understand our data, and make decisions? As my background is in plant biology and crop protection, I use examples from the agricultural sciences. The biostatistical tests, approximations, and rules of thumb described are, however, applicable to many other branches of biological experimentation. Finally, the book, which contains the contents of this course as well as additional material, is available to you as supplementary reading in the form of a free downloadable PDF. In the field of agrochemical research and development, statistical rules of thumb are general guidelines or principles that can be used to analyze and interpret data. These rules are based on statistical theory and experience, and they can help researchers make sense of complex data sets and to draw meaningful conclusions. Statistical rules of thumb are useful because they provide a quick and easy way to evaluate the quality and reliability of data, to identify patterns and trends, and to communicate results to others. In this course, we will explore the advantages and limitations of these rules, and we'll consider the factors that influence their usefulness and applicability. We'll also discuss the challenges and considerations that are involved in using statistical rules of thumb in agrochemical R&D, and we'll consider the role of these rules in the decision-making process. This course comprises 4 essential statistics modules which lead on from each other, beginning with proposing a hypothesis, moving on to designing and implementing experiments. Using the information we have learned there, we move to descriptive statistics, and finally to inferential statistics. In the module proposing a hypothesis, the key objective is to provide students with a solid understanding of hypothesis testing and the potential errors associated with it before embarking on experimental design. Students will learn how to formulate research questions and hypotheses and define null and alternative hypotheses. By the end of this module, students will be able to critically evaluate and develop a clear understanding of the potential errors that may arise during hypothesis testing. They will gain the necessary skills to propose well-designed experiments based on sound statistical principles. Designing and implementing experiments, significance, power, and effect. The objective of this module is to equip students with the knowledge and skills to design and implement experiments effectively. Students will explore the concepts of significance, power, and effect size and understand their importance in experimental design. They will learn techniques for determining the appropriate sample size, randomization, and blocking methods to reduce bias and increase the validity of experimental results. By the end of this module, students will be able to design experiments that are statistically rigorous and yield reliable and meaningful conclusions. From this, we move on to descriptive statistics, critically evaluating experimental data. In this module, the focus is on the critical evaluation of experimental data using descriptive statistics. Students will learn how to summarize and interpret data. They will explore data visualization techniques to effectively communicate and present experimental findings. The key takeaway from this module is that students will develop the skills to assess the quality and reliability of experimental data, enabling them to draw meaningful insights and make informed decisions based on these results. Finally, inferential statistics, accepting or rejecting the hypothesis. The objective of this module is to provide students with a comprehensive understanding of the inferential statistics and their role in accepting or rejecting hypotheses. Students will learn about data types, assumptions for commonly used parametric hypothesis tests, data transformation, and the interpretation of p-values. By the end of this module, students will be able to make informed decisions about accepting or rejecting hypotheses based on statistical evidence and understanding the limitations of such conclusions. In addition to the statistical modules outlined above, this course covers additional modules related to trial data evaluation, including corrected efficacy, area under the disease pressure curve or AUDPC, dose response evaluation, and synergy calculation. Topics not always covered in introductory biostatistic courses, but which are essential elements of the trial management toolbox. Corrected efficacy covers the isolation of efficacy attributable to the pesticide treatment. This module focuses on isolating the efficacy attributable to the pesticide treatment in agricultural experiments. Students will learn statistical approaches to adjust for responses occurring independently of the treatments at zero dose or untreated controls, and accurately estimate treatment effects. They will gain an understanding of how to interpret and report corrected efficacy results, ensuring accurate assessment and comparison of different treatments. The key takeaway from this module is the ability to distinguish and quantify the true efficacy of a pesticide treatment from other factors that may influence the experimental outcome. Area under the disease progress curve, or AUDPC. The objective of this module is to equip students with the skills to combine multiple observations from disease progress experiments into a single cumulative value using area under the disease progress curve. Students will learn how to assess disease progression over time and quantify the overall disease impact. By the end of this module, students will understand the significance of AUDPC in disease management and be able to effectively analyze and interpret disease progress data. Dose response, predicting values from measured values. In this module, students will delve into dose response analysis, enabling them to predict values based on measured data. They will learn how to model dose-response relationships, estimate effective dose levels, and assess toxicity and response relationships. By the end of this module, students will have the ability to predict and interpret responses to varying doses accurately. This knowledge will be crucial in determining optimal dosage levels and ensuring the effectiveness and safety of treatments. Synergy. This final module focuses on the concept of synergy and its applications in extending the patent life of mixture partners and defending market position by reducing active ingredient rates. Students will explore statistical approaches to measure and optimize synergy effects, enabling them to maximize the benefits of mixture treatments. By the end of this module, students will understand how to assess and report the advantages of synergistic treatments and make informed decisions to enhance market competitiveness while reducing the rates of active ingredients.